Hi, everybody. We're going to get started now. Thank you all for showing up, and thanks everyone who's online. I'm really excited that we were able to invite Dr. Corey Krabbenhoft out here to come give a talk. I'm excited for a couple of reasons. Number one, she's a fish ecologist, which we don't really hear a lot from, so I thought it would be really a good way to round out the seminar series by inviting her. Corey also has a really interesting career trajectory. She is from New Mexico and started her fisheries career there, working in desert streams. I actually met Corey in, I think it was like January or February of 2014, right during one of our polar vortices. So she came directly from New Mexico to that, and then decided to do her PhD in Detroit at Wayne State, where we were in Donna Cashian's lab together. And now Corey is out at University of Buffalo, where she is an assistant professor working on a whole host of really cool stuff, including lake trout around Goby, native mussels, and she also knows how to ride a unicycle. So let's welcome Dr. Corey Krabbenhoft to our seminar series.
If anybody wants tips on unicycle riding, I'd be happy to discuss that afterwards. But thank you, Anna, for that lovely introduction. That could have been much worse, so I appreciate it. And thank you all for being here today, as well as those of you joining us online. We appreciate your interest, and it's been a lovely morning here at your facility, chatting with everybody. You're all doing amazing, very cool work, and I'm so excited to follow up with some of you about that. So I normally start these talks with why should we care about the Great Lakes and those sorts of things. So I probably don't need to harp on that topic too much for the sake of this audience, but these are the things that I normally like to tell people about our fisheries and the economic and the cultural value. And so when I finished my dissertation work here in Michigan and then eventually made my way over to Buffalo, I was excited to sort of stay in the realm of the Great Lakes, just because there's so much enthusiasm and so much amazing work going on in these systems, a lot of which I got to hear about today, and many of which you all are directly involved in. So when I started my lab at the University of Buffalo, the sort of unifying theme in a very general sense was aquatic ecology. And a lot of what I'm going to be talking about today, I have to give credit to my amazing students, 3 of whom are pictured here, and I'll introduce them a little bit when we get to their specific projects. But the way we sort of approach things in my lab is with a very big emphasis on sort of the organismal biology and how those organisms function in their environments. And so we're interested in the stressors that these aquatic communities incur, both physical and biological. We sort of look at this across maybe more focus on like population and community levels, but we're also interested in a number of different spatial scales where we're looking at what's going on in a single reach of a single stream, as well as some of the patterns that we see across much larger scales like the entirety of Lake Erie. So as we sort of move through, and I've been developing my lab, and we sort of move through the kind of work that we've got going here, one of the things that really excites me about working in the Great Lakes specifically is all of the amazing biodiversity. And as Anna mentioned, my background is largely in fish biology, though I've dabbled here and there in other things. There are a lot of really exciting and interesting questions that can be asked about the fishes in the Great Lakes and their biology and what's going on as they interact with one another and their environments. And so the way I sort of approach my thoughts about how these interactions work is largely through like a basis in some theoretical ecology. And so just to give us a brief refresher from our intro bio days, one of the sort of key core concepts that I'm excited about is dynamic energy budget theory, which I won't bore you with all of the details, but basically this is how an organism uses its energy, right? So it's got a bunch of very basic biological functions that it needs to perform, and how well it does those things, how efficient it does those things, how well we're moving forward into future generations of that population, all sort of boils down to these fundamental processes. So we have ingestion, we consume things from our environment, we're concerned about what's going on in the food webs of these organisms. All of that energy is either assimilated into their tissues or excreted, and there's nutrient implications for that. We can think about the production both of our somatic tissues or into our gametes, so both in terms of building our physical body structure, and there's interesting things about body size that we can ask questions about, and the maintenance of those structures over time. And then we can also think about how we reproduce and grow the next population and how we invest in those things. And I think this is sort of a nice way to sort of think about what is an organism and why is it important and why does it do all of the things that it does, because a lot of what we do in my lab really focuses on a number of these very basic processes and thinking about how organisms function in their environment relative to these sort of core biological functions. And so the talk that I'm going to give today is going to cover two different projects that we have going in my lab, largely with respect to the growth and maintenance of tissues and interactions with other organisms in the food web, and then one will focus specifically on their reproductive output. And so the study organisms that I'm going to be talking about today are the Lake Erie, are the lake trout, which we're specifically focused on the population in Lake Erie, and which is our sort of native half of the talk, and then we're going to move to invasive species that are now occupying the Great Lakes and talk a little bit about round goby invasion. So we'll start with our lake trout story. Now, as I mentioned, this is going to focus specifically on the population that we find in Lake Erie, which is viewed here. It's an interesting story. For those of you who don't know, I won't harp on because I know we all have quite a bit of background, but just to get us on the same page about lake trout specifically, they had a bit of a struggle in the 1900s in terms of their population sustainability. There was a long period of overfishing, which I think was relatively ubiquitous across all of the lakes, but there's also habitat degradation, so water quality, and all of these things that we sort of think about in terms of maintaining the ecology of these ecosystems. Were having a bad time, and but lake trout were also particularly impacted by the introduction of non-native species. So there are stories about their interactions with alewife and smelt, and particularly sea lamprey, lake trout being one of the fishes that sea lamprey parasitize at a relatively high rate. So there was a lot of sort of multiple stressors affecting these lake trout populations that ultimately resulted in their extirpation from Lake Erie in the 1960s. This was a very extreme example of these population changes. There were similar declines in the other Great Lakes, but it was most pronounced in Lake Erie. I think in part because of the sort of unique nature of Lake Erie being relatively shallow. The temperature dynamics are much different than Lake Superior as an example, but it's also very much at the southern end of the range of lake trout. So the lake trout natural range extends really far into northern Canada, and they're sort of thought of as a sort of cool water species. And so Lake Erie is already at the southern extent of their range, and then when you add all of these other environmental degradation factors and invasive species, it sort of was a very bad combination of events for lake trout. So they were extirpated in the 1960s. And in the 1980s, as a response to this, there was a concerted focus on rehabilitation of lake trout in Lake Erie. So the Lake Trout Task Group was sort of charged with rehabilitating the Lake Erie lake trout population, which largely centered around stocking from hatcheries. So lake trout were raised in hatcheries. Hundreds of thousands of them were stocked into Lake Erie every year. That was met with a variety of successes, but it sort of came at the end of an investment in environmental improvement in the 1970s when we had the Clean Water Act, the Endangered Species Act. All of this sort of investment in environmental improvements happened in the 1970s. In the 1980s, we started talking about reintroduction of lake trout into Lake Erie. There was a sort of a period of trial and error where they were trying different things in terms of how they stocked these lake trout populations. But the real sort of success started to happen after they combined that with lampricide treatment to bring down the rate of lamprey parasitism on lake trout, which we're seeing here in this photo on the right. And the figure I'm showing here on the left, this is year on the bottom. I apologize for the size of the text. Basically, we're going from 1980 to 2022. And here we're looking at basically the rate of lamprey parasitism. And you can see prior to around 1986, 1987, the rate of wounding from lamprey parasite wounds on lake trout was much higher, and it's sort of reached a sustainable, relatively low level. And so the combination of the concerted and consistent hatchery reintroduction of lake trout and the sort of dampening of the wounds incurred from lampreys have really helped lake trout reintroduction, and we now have what's considered a relatively healthy lake trout population in Lake Erie today. Part of the interesting aspect of this, though, is that it's still entirely maintained, as far as we understand, from hatchery stocking. So we continue stocking today, and it's not what we would call a self-sustaining population. And one of the interesting things about the method that we went about in terms of restocking these lake trout was that they were coming from all of these other stocks that were not native to Lake Erie itself. And so there's 6 of them are shown here. That had varying levels of success when those strains were stocked into Lake Erie. So the Finger Lake strain, which is this sort of light blue color here, has largely been viewed as one of the more successful. And so over time, as we've developed this lake trout hatchery rearing program, more effort has been put into that Finger Lake strain because they've been the most successful in occupying the habitats in Lake Erie. And so it's not entirely clear what the difference is between these strains and why they're responding to these habitats in such a different way. One of them we think is because the Finger Lakes have sort of co-evolved more so than with sea lamprey than some of these other stocks, so they might be better behaviorally at avoiding lamprey wounding, since we know that lamprey wounding is a particularly problematic problematic occurrence for the maintenance of lake trout. So the sort of hatchery history of lake trout in Lake Erie has encountered struggles along the way, but is resulted in relatively healthy population at present. And the sort of ultimate goal of the rehabilitation program would be to get us to a place where we no longer have to stock from hatcheries, obviously. Can we get a self-sustaining population of lake trout in Lake Erie? So there's a number of folks across various state and Canadian entities that go out and monitor the situation with lake trout in Lake Erie, including these annual assessment surveys. But interestingly, we have in the past couple decades found actual evidence that they might be reproducing in Lake Erie. And this is the first time this would have happened in decades since their extirpation in the 1960s. It's at a very, very low rate as far as we can tell, but we think that reproduction is happening. So there have been surveys that go out and regularly catch lake trout. The ways that the hatchery reared individuals are marked so that we can keep track of which ones are from the hatchery. There's 2 methods so that if there's error, we can sort of keep track. One of them is a cutting of the adipose fin, and the other is a coded wire tag that keeps track of the individual fish. And so individuals that are missing both of these tracking methods are considered potentially wild. But that's less than 1% of the annual assessment surveys. But there was some evidence in 2004 from Ludson et al., where they used otolith microchemistry to go in and evaluate a handful of these fish to determine whether they had signatures in their otoliths that would suggest they came from a hatchery as opposed to being reared in the lake. There was some evidence. They had 4 out of 28, so still relatively small percentages. But this is sort of good information and sort of good evidence that we think that there might be some level of wild reproduction happening in this previously extirpated species. There are a couple of folks who've gone out and sort of started looking for where this might be happening and under what conditions that might be happening. There was some egg sampling where they went and took hatchery origin lake trout to see if they could raise the eggs in a natural environment. There was some sampling for egg deposition in various parts of the lake, but they were determined to not be viable. But there were ripe fish caught in nearshore gill nets, so they were ready to spawn. So there's been sort of tangential evidence, but nothing that sort of solidifies this potential wild reproductive ecology. And there's been a handful of recent papers that sort of looked at this spatial ecology of lake trout in Lake Erie specifically to see what we might be considering spawning behaviors, to see if we can sort of identify where this might be happening, to do a better job of surveying for evidence that actual reproduction is taking place. So in 2021, I believe, the very first effort to locate a spawning reef for lake trout occurred. This is just off the waters of the New York and Pennsylvania shores of Lake Erie, where there are these historical sort of where we know that historic populations of lake trout had spawned. This is essentially an offshore reef that's composed of cobble and rocks that make up the primary spawning habitat for Lake Erie, which we know from other systems. They went out and used acoustic telemetry from the GLATOS network to sort of identify these hotspots of where aggregation was happening during the known spawning periods of lake trout, which tends to happen from October to December. The coloration that you're seeing here in these plots is the density of the individual fish that are congregating there during that time. And they went out and used fry traps along all of these positions here in the circles to determine whether or not there were actual larval fish being produced. Which they found. Here's a larval lake trout here, pictured here on the bottom left. And so this was the first actual hard evidence that lake trout were actually spawning in Lake Erie for the first time in decades, which is really exciting. But this was sort of an exploratory first effort of the spawning behavior and the locations. And so the project that I'll be talking about today is really sort of taking the next steps, building on that paper by Jim Markham and all. Where we want to understand what the environmental conditions are that are resulting in a positive production of lake trout larvae, and what are the sort of environmental and trait-based conditions of lake trout that are sort of making that happen. And I do want to briefly give a shout out to Susanna Kyleg, who's a PhD candidate in my lab, who is basically doing all the work that I'm up here taking credit for today. So the things that we're excited about for lake trout and what we think we know from other systems in terms of what's going to be important in determining when and where and why they're reproducing. There's individual traits that we think might be indicative of their spawning behaviors and their relative reproductive success. So the strains that I talked about, the different hatchery strains that are actually being put out into the lake, as well as the differences between their behavior in terms of males versus females, as well as their body size. And so to give you an idea of what I mean about this diagram here on the right is sort of illustrating the spawning behaviors that are different between sexes. So here we see a male aggregation over a spawning shoal. Males essentially arrive to the spawning area first. They sort of scope out what's going on. The females come later, and there's this courtship behavior that happens. So it's called finning, where they sort of come up to the surface and they're sort of exposing themselves and jumping around a little bit. And then there's actual spawning that happens. And so these are all sort of clues and differences in behavior that we can use telemetry information to sort of track over time. But we also think that the environmental conditions at particular times of year, and particularly over the spawning shoals, is going to be important in determining whether or not reproduction is going to happen and whether or not it's going to be successful. And so we're looking into these environmental drivers, including water temperature, the sort of wind and storm action, as well as the photo and diel periods associated with those behaviors. This is what a spawning reef actually looks like. So it's we know from other systems that they're lithophilic spawners. So they're spawning over tops of these reefs. All the eggs sort of settle into the interstitial spaces between these rocks where they're relatively protected from being blown out and potentially from predation. So we think that flow is an important part of this, but also the slope of the shoal itself, the depth where that reef takes place, and then the water currents that are sort of maintaining the conditions on that reef. So one of the things that we wanted to consider when we sort of decided we wanted to figure out what the bottleneck is, right? So lake trout might be reproducing, but they're certainly not at a sustainable level. So there's some sort of bottleneck happening in that population. We know that they're reproducing, but where is the problem occurring? So if we remind ourselves of what lake trout life history looks like, we start with eggs. The spawning activities happen in October to December. They sort of overwinter on those spawning reefs. And in the spring, in say March-ish, they're going to start hatching out into larval fish. There's very little known about what happens to the sort of juvenile life history stage in Lake Erie. It's sort of an ongoing mystery where the fisheries biologists sort of lose track of them for a few years. And then they start to reproduce at a few years of age, so moving back over to those spawning reefs. So for the purposes of our particular study and wanting to understand their reproductive ecology and what's driving those events, we're going to focus on this sort of early part of these life history stages. So I sort of briefly mentioned that we're tapping into the Great Lakes Acoustic Telemetry Network for this, or the GLATOS. This is an illustration of the lake-wide network where we have all of these buoys out in the lake that are picking up the pings from fish that have been surgically implanted with a transmitter. We can use this to keep track of where the fish are moving in the lake at different periods over the course of the year, so looking at seasonal differences and specifically focusing in on those spawning times. We know from that Markham paper that a lot of them, and the funnel paper that I showed from Tyler Funnel et al., that lake trout are largely congregating in the eastern part of Lake Erie during spawning season. And so as a result of those initial findings, there were 3 additional acoustic telemetry arrays that were set up along the southeastern shore on the shores of New York and Pennsylvania in Lake Erie. They were set out in 2018, 2019, and 2020, specifically during spawning season, so from October to December. These are finer scale arrays than exist in the normal GLATOS network, so the resolution of those movements is much finer, much higher resolution than what we get from the lake-wide GLATOS receiver network. So these were set out in the fall during spawning time, and we had a total of 287 lake trout were picked up in those arrays. And the reason they were sort of put here is because this is where the hypothesized aggregations were happening during that reproductive time. We're currently expanding on the initial findings from the Markham paper, where we're going out and doing additional fry traps. This is an update of those from those very fine scale receiver networks, where the color, more yellow color, indicates aggregation during that spawning time. So you can see these sort of spawning shoals similar to the Markham paper. And the locations that are marked here in stars are where we've pulled fry traps that actively have larval lake trout in them. So we're sort of doing a proof of concept with the fry traps to make sure that the conclusions we're drawing from the telemetry data are actually resulting in production of larval fish. To sort of take the Markham methods one step farther, we're looking into sort of a trait-based analysis of all of these fish, in addition to the physical habitat that they're using. So we have the strain from all of the individuals that were surgically implanted with transmitters. We also have a fin clip that we can use to genetically determine the strain. So which hatchery strain and hatchery origin did it come from. Those were largely determined by U.S. Fish and Wildlife and the Ontario Ministry of Natural Resources. We're additionally in-house doing sex identifications genetically using PCR and gel electrophoresis for the SDY gene, or sexually dimorphic on the Y chromosome, which has been previously, markers for which have been previously developed to determine sex in lake trout and other salmonid species. And then we're additionally doing some evaluations of predicted weight based on the size at which these fish were when they got tagged and were released to what we expect they would be now. So we're using all of these sort of trait-based assessments to learn something about whether or not there are differences in reproductive output based on all of these individual traits. And here's some preliminary data that I'll show you, where the top figure is showing the 5 different strains of lake trout, so those hatchery origins. We have very different results based on their strain assignment and in terms of how long they're hanging out on these spawning reefs. There's also but I should note that there's also very wide sample sizes for all of these strains, where the Seneca Lake strain, this second from the right here, is by far the most abundant that we're seeing. But there are different amounts of times that they're spending on these spawning reefs, indicating there could be some sort of different habitat preferences relative to these hatchery strains. The figure on the bottom here is showing us the relative timing between male and female arrival on these spawning reefs, and then again relative to their size. So we have small males and small females showing these first 2 lines here, and we have large males and large females, suggesting that not only is the arrival of male and female different on these spawning reefs, but also that can be sort of affected by the size of the fish, where small individuals may be arriving earlier, larger individuals may be sort of developing their gametes for longer and showing up later. So there could be, depending on the environmental conditions, differential reproductive output based on those individual fish traits. So we're combining what we found with the individual fish traits with the environmental conditions that sort of lead up to the spawning event in the first place. So the idea here is to determine whether or not we can figure out what the drivers are so that we can better predict when and where spawning might be happening. We are getting water temperature from the receiver loggers where these telemetry arrays have been put out. And we were additionally using weather stations and buoys from NOAA. Thank you all very much for maintaining these things to look at wind and cloud cover data associated with the timing of these spawning events. The top figure that I'm showing you here is relative to water temperature on the x-axis and the number of fish that we have moving into our fine scale arrays as a result of that. Unsurprisingly, there's some maybe correlation with this where they're hanging out. 10 degrees is usually cited as sort of when we expect we're going to start seeing spawning activity. So each one of these dots represents an individual fish, and we're sort of seeing that taper off as we get into warmer environments. And then the figure here on the bottom is relative to wind speed. So the vast majority of the fish are hanging out over atop these spawning reefs when wind speeds are relatively low. And we are starting to look into gust speeds as well, which we think might be helping sort of clean the sediment out from these when we have these big isolated wind events. Then some of these fish might be moving into the spawning reef shortly thereafter. Interestingly, the water temperature, as you might predict, correlates nicely with sort of that turnover that we see in the fall, where we're seeing the colder water sort of at the top as fall starts to set in, and then we can nicely see the period of turnover. So that's when we're starting to see a pickup in some of these spawning behaviors. And when we think about the wind speed, we're also looking into, to the extent that we have the detail to do so, the bathymetry of where these arrays were placed and how that corresponds to what we expect wave action to be doing as a result of these wind speed drivers. But I did have some very productive conversations with folks this morning where we might be able to get into this in a little more detail. And the idea here being that these sort of big wave events or big storm events might be coming and blowing all the sediment out of these spawning reefs such that all of the interstitial spaces are then free for the eggs to move in. So we'll be looking into that into a little more detail as we go forward. The next phase of this project is going to be to develop a habitat suitability index for lake trout reproduction in Lake Erie. And our first effort to that end is to do a more detailed job of characterizing what the habitat looks like on these rocky reefs where they're going to spawn. We started last summer, and we're going to continue this summer, where we go and do side scan sonar above these spawning reefs, and we're going to also be doing some drop down quadrat surveys to sort of have a camera visual to sort of ground truth what we're seeing in terms of the side scan sonar. This will help us really nail down that sort of the orientation of that spawning reef, the slope, all the depth, all of those habitat details that we're then going to be able to sort of pair with the behavioral information we got from the telemetry approaches. I'm also interested in looking in greater detail to how the hydrodynamics are affecting what's going on. We briefly sort of have some anecdotal information that this is going to be important, particularly in terms of keeping sediment out, but I'm interested in pairing this with some hydrodynamic modeling and potentially going out and putting sensors on the reef itself to determine whether either flows passing over the reef or hypolentic interactions are important in keeping these eggs in good condition, particularly since they're going to be on these reefs for several months and need to sort of make it through the winter in order to result in adequate lake trout reproduction. So right now, this is an ongoing project. We have more field work has already started for this summer. Mostly what we're finding right now is that there are some spawning conditions that we think are important that have resulted in this sort of new wave of lake trout reproduction. That includes the sort of wind and movements that we've seen over the areas where we have these spawning reefs, but there's also some individual like trait-based factors that are important in regulating what's going on. The next step will be sort of filling out that habitat suitability that will better understand not only what habitat they prefer to spawn on, but what's actually resulting in the development of the lake trout larvae in the end. So whether or not there are potential habitat sinks that are not resulting in high enough hatch rates. the sort of ultimate big picture goal would be to sort of work toward a set of conditions that we think are productive for lake trout sustainability, and then be able to apply that to other areas of Lake Erie. Because right now, these are sort of the only areas of the lake that we are focusing on in terms of having a lot of information about what's going on with reproduction. We suspect that there are probably others, but we don't really know where to go look. So that'll be sort of the long-term goal for this project as we move forward. So that's my first spiel on one of our Great Lakes native fishes on lake trout, and thinking about habitat and how it interacts with its environment and how that leads to reproduction. But as I mentioned, I'm also interested in species interactions, and one of the great ways of doing that in the Great Lakes especially is to look at the invasion of various species, and we've got a lot. So I'm going to dial into one example, and we're going to be talking about the round goby. So again, to get everybody on the same page, probably everybody in the audience knows this story at least a little. Round goby were brought over in ballast water from Eastern Europe, Black and Caspian Seas, released into the Great Lakes. They were first sampled in Lake St. Clair in 1990, and as this sort of range map here shows you, very quickly radiated across all 5 of the Great Lakes within just a few years, and are now making it up into inland waters, into inland lakes and a variety of tributaries. It's been a very prolific invader. It's been very successful. There's a lot of examples of how it interacts with fishes and other organisms in the lakes themselves, and there's an increasing amount of interest in what's going on in our tributary systems. Particularly because we've had such great help in moving them around. So not only do we have sort of the freight and cargo routes are moving things around, but we've got a sort of population density of people who are sometimes using them for bait and moving them around our Great Lakes watersheds. So it's been a really successful initial invasion, but also the sort of secondary spread as it sort of moves across this area of North America has been particularly prolific. The impacts that we know that round goby has on a number of systems are relatively diverse. It does show up in the diet items of a number of larger piscivorous species in the Great Lakes, but some of these smaller bodied species are relatively negatively impacted, largely through competition. Diet overlap is sort of a key example where there's evidence that they're competing for similar diet items with some of these smaller species, particularly the darter species that are relatively similar in body size, morphology, and habitat. It does have, round goby do have impacts on other fishes, for example, through egg consumption, which is one way it's maybe potentially linked to our lake trout problem, where all of these spawning reefs are covered in round goby that are then eating all of the eggs. We have seen examples of egg consumption in tributaries as well, but it's also serving as a vector for avian botulism. So there are other negative impacts beyond it serving as a prey item for some of our larger species. But this sort of diet overlap and competition with these other native benthic species is an area of interest as we move from lake to tributary systems, because the populations of those kinds of fishes are much higher. And in some areas in the region, we do have some that are of conservation concern for a variety of reasons. So the sort of overarching question for this inquiry, this line of inquiry, is about what the ongoing impact of round goby invasion is in the Great Lakes as we sort of move from this lake to these tributary systems. And I'm particularly excited in using round goby as sort of a test case for some of these basic questions about the processes of invasion and invasion biology in general, because it has occurred in so many systems. There are lots of sort of replicates that we can use to understand some of these larger patterns. So again, I'll give some credit to some of my students here. This is Matt Scott, who just defended his master's on Monday, and Catherine Clark, who's an undergraduate in the lab, who will also be graduating this semester, sadly. They went out and did a bunch of field work this last summer in Tonawanda and Ellicott Creeks, which were here in western New York State, snuggled between Lake Erie and Lake Ontario. Ellicott and Tonawanda Creek run into the Niagara River, so they're connected to the Great Lakes through that connecting water. And then Buffalo Creek is another creek that runs into Lake Erie directly. Interestingly, these were sort of our sample sites, which we targeted because we knew some of them had round goby, but we think we caught the invasion front in 2 of these, where these bottom 3 sites in Tonawanda Creek had round goby in them. This 4th upstream site in Tonawanda did not, but there's this sort of large natural log jam happening there that we think is physically keeping them out of the upper reaches for the time being. In Ellicott, they've moved up to this 3rd site in Ellicott as well, but there's a natural waterfall that occurs there that's not passable by round goby. So this 4th upstream site in Ellicott Creek is not yet invaded until somebody comes in with a bait bucket and moves them around to the impoundment. And Buffalo Creek has a series of low head dams. It sort of serves as our natural control because it's not invaded at all. So we have sort of a nice representation of the progress of invasion in these sample sites. But one thing that's I find really interesting about the process of invasion in these rivers is that it's essentially happening in a linear direction, where if you have a terrestrial system and an invader comes in, they can move out in all directions. And if you have a lake system and you have an invader coming in, there's lots of like 3-dimensional habitats that they can occupy and move into. Whereas the direction, we know the direction of the invasion in a river because the source population in this case is the Great Lakes, and they're actively moving upstream. So we know the direction of the invasion. And so one of the things we're doing here is sort of using a space for time substitution, because we don't have a lot of old data on these sites in terms of when these invasions happened, but we know that the upstream sites are most likely the most recently invaded. So this is just sort of a brief overview of the types of fish assemblages that are in these creeks. It's about what you would expect, very heavy on our minnows. There's some suckers. The blue here represent our darter species, which we're particularly interested in for the purposes of their interactions with round goby. And you'll see that round goby in the black here occupies some of these sites. The diversity is not terribly different between all of these sites, and we had relatively similar habitat values. We did additional like habitat and water quality assessments as we went out. So they're relatively similar in a lot of aspects, with the exception of that sort of longitudinal factor in terms of the invasion direction. So since one of the main ways we know that round goby impacts native fish species is through this diet overlap and potential competition, we're using a food web approach and using stable isotopes. Just as a refresher for those of you who haven't heard about this in a while, we're largely using carbon and nitrogen isotopes to sort of help us reconstruct what the structure of the food web looks like. Carbon is largely used to determine the difference between primary producers. So where is the energy coming from in this food web? In streams, we can often use this to delineate in-stream resources like algae from terrestrially derived resources like detritus, leaf litter, that kind of thing. And then as we move up in our nitrogen dimension, this sort of follows the trophic structure up, where every time we move up a trophic level, we get higher nitrogen signatures. So we have our primary consumers at the next level, then our intermediate predators, our small fish, our predatory invertebrates, and then our top predator. So we can go and take tissues from all of these organisms in our study system, and then use the relative abundances of carbon and nitrogen to reconstruct what the basal resource is for each of those diet items and the sort of the community as a whole, and what trophic level they're occupying. So from all of our sampling that we did this last summer, this is all of the data put together, which is a lot, so we'll break it down here in a minute, with all of our fish up here in red, the invertebrates community in green, and then all of the plants occupying this blue polygon here. So we're basically sampling every level of this food web, and then doing our best to sort of attempt to reconstruct who's eating who and where the energy is coming from. And there's a couple of things that we can do with this type of information. One of them is to use stable isotope mixing models to reconstruct what we think all of these fish are actually eating. So if we hypothesize that there's competition for resources, we can reconstruct what those resources are. Then we can look at the niche breadth in this biplot space, where a larger ellipse here around these points is going to suggest that they're eating a really diverse set of things. And that can correspond to niche breadth, or the diversity of the things that they have in their diet. And so we're using Bayesian models to reconstruct that. We can also look at the relative trophic position based on the change from each trophic level to see if that's shifting over time or in space. So let's look at some of this. I'm going to start by looking at the results from our mixing models, so reconstructing what's actually in the diet of these fishes. And I'm going to show you a subset of this from our Johnny darters here on the left to our round gobies on the right. All of the prey items are over here on the right. You don't necessarily need to know what those are, but for those of you who enjoy a good macroinvertebrate info, there it is. Okay, so at our downstream, I'm just going to look at one of our creeks. This is Tonawanda Creek that we're using it as an example. At our downstream site, again, the sort of oldest invasion in this system, which in this case we know is at least 15 years at a minimum old. We're seeing Johnny Darter has relatively few diet items, so does round goby. There's not a ton of overlap in terms of like the similarity of what they're eating. So there seems to be some sort of niche partitioning that has happened in a really established invasion. If we go mid-invasion, moving upstream, again, to a more recently invaded site, there's very, very little diversity in the Johnny darter gut especially. There's not a whole lot of overlap, but both the darter and the goby have relatively simple diets. But at our new invasion that's only been what we think is in the last couple of years, both of them have a much more diverse diet overall. But you'll also notice that the corresponding colors here sort of indicate that there's quite a bit of overlap in the individual items that they're eating. And so what we think is happening is that that there's some sort of temporal factor associated with the impact of this invasion, where there's a relatively high diet diversity for both individuals early on. They potentially are in really high competition and sort of moving away from that over time till we get to sort of a new equilibrium where they're eating different things, but they've adequately partitioned the diet space. And if we look at an uninvaded system from our Buffalo Creek sample site, this is Johnny darter and rainbow darter, they have relatively diverse diets. So this sort of informs that we think that this is sort of the natural state before invasion happens, where we have really high diet diversity early on. And then if we look at the sort of proportion of the overall, what are they eating in terms of the overlap of those ellipses, what we're looking at here is the proportion of the overlap between our darter species and our round goby at each of these sites. And here we're listing them from the sort of upstream sites on the right to the downstream sites on the left. And what we're seeing is that there's a much higher instance of overlap between those individuals at more recently invaded sites that then diminishes essentially over time, but in this case, space. And we can also look at that in terms of the proportional abundance of the round goby. So at a place where the invasion has expanded, there's a lot of individuals, we tend to see the proportional niche overlap between our native and invasive species is much higher. And again, these are happening at the most recently invaded sites. So because we have this sort of longitudinal upstream invasion and this space for time substitution that we're working with, it's interesting to see this sort of trophic overlap become an issue. Because if we then look at sort of the overall diversity, that ellipse area, specifically for the darters, if we look at that over the course of proportional abundance of our round goby, they tend to decline in terms of the diversity of what they're eating as round goby become more abundant. So there's a decline in their niche breadth as round goby become more established and more abundant. And what we're seeing in terms of their trophic position, if we look at the darters here in this pink color and round goby in the green color, they tend to overlap pretty well in terms of their trophic position when round goby first move in. But as round goby become more abundant, we start to see this separation such that there appears to be a decrease in that overlap with round goby as that population becomes more established, in part because they're shifting where they are in their trophic position. So they're moving to a different trophic level to avoid competition with round goby, we think. If we actually take a little bit more detailed look and look at the gut contents, the physical contents of the round goby diet, this is brand new data, so I haven't totally spent a ton of time looking at it, but here we're looking at essentially a one-to-one line of the invertebrate community from an actual macroinvertebrate sample compared to what we're finding in round goby gut contents. And it turns out that there's a lot of stuff that they're not eating, so they appear to be relatively picky, which is different than what our expectations are based on what we know from round goby. invasions in other places, where if they're inside of this sort of like gray band, they're eating those things in relatively about the proportion they exist in the environment. So relatively opportunistic feeding strategy. Everything above that line is something that the round goby is eating in a greater abundance than it actually exists, so something they might be preferred. Everything below that line is things that they are potentially avoiding. So we see very, very specific feeding strategies here according to a handful of these invertebrates. I have yet to sort of sit down and figure out what's going on here in terms of how this corresponds with our stable isotope data, but we think that this is going to be sort of a useful way to connect what we think those diet item assignments are, and if we're missing anything, we can potentially pull that out here in our gut content information. But one of the interesting things that we've come up with from the gut content data so far is that if we plot each of these round gobies, each one being an individual fish, and rank them using an electivity index from being a relative generalist, eating things that they at the rate that they encounter them, to a specialist, we see a lot of them are really, really specializing on a handful of things, but it varies according to the size of the individual fish, which we know there are ontogenetic shifts in round goby foraging from their native range, because they do eat bivalves in their native range and they need to be a certain size in order to be able to crunch the shells. So there is some sort of component of the actual size of the fish in terms of what they're eating. So we're interested in looking at the relationship between that sort of feeding strategy in terms of specialists versus generalists, especially as it relates to the stage of invasion and the size of the fish. So we just started digging into that, and there will be more to come. But our sort of immediate takeaways is that we're definitely seeing differences in the food web structure of these invasions at what is purportedly the invasion front versus a well-established invasion much farther downstream. We think that competition may be the highest between native individuals at that invasion front than it is at a more established population, but that there might be some of this like boom and bust where they come in and they're very abundant and lots of bad things are happening, and there's some sort of adjustment period over time where things settle down a little bit, and there might be some niche partitioning to sort of help compensate for that new individual in that community food web. This is particularly important because there are areas that have yet to be invaded by round goby that we think that they're going to be in the near future. One of these in New York is in Lake Champlain on the eastern side of the state, so outside of our Great Lakes Basin. But round goby are knocking on the door of Lake Champlain, and folks are very concerned about that for a number of reasons, particularly the fishes of Lake Champlain itself, but there are also a number of endangered darter species in the tributaries to Lake Champlain. So it's something that folks are very concerned about. We did some sampling there last summer, which we hope doesn't have to be used as a before data set, but we have just in case. So that's something that folks are keeping a close eye on with respect to the ongoing spread of round goby. So overall, the sort of two stories that I've chosen to share with you today, I hope are interesting from the fisheries biology context. One has to do sort of with the environmental drivers of, the long-term population dynamics of a native fish. The other being sort of these species level interactions with an invasion that's ongoing in the Great Lakes and outside, and the overall impacts on the aquatic communities. But we're excited about these kinds of things in the lab, and we hope that these data can be useful and informative in terms of how we go about managing the Great Lakes and these aquatic ecosystems, both in terms of their sort of natural ecological integrity, but also how people interact with the fisheries and the water quality and those kinds of things. So I want to thank you again for tolerating this very brief overview of some of the things that we're doing. It's been absolutely wonderful to be invited here to speak with you today. I definitely need to acknowledge the students who did a bunch of the work that I shared with you, Susanna Keilig, Matt Scott, and Catherine Clark, as well as some of our partners and funding resources, the State Water Resources Institute, as well as the folks at the New York State Department of Environmental Conservation, who are generously letting us use their boat time and their telemetry information, and we're working very closely with them. There's a number of other folks from the lab and UB and other collaborators that are listed here. I'm sure I've missed some, but they've all been instrumental in what I've showed you today. And if anybody wants to chat about anything that I have talked about today, either here or in the internet land, this is my email. You're more than welcome to reach out. And lastly, I'd like to thank my colleague Anna Bageld for inviting me here today. It's been a pleasure, and if we have time for questions, I'd be happy to take some.
There's one online.
I can repeat questions too, if that's easier.
So yeah, any questions in here? Oh.
We're passing around a microphone for those of you on the internet.
Hi, Corey. Thanks. Great presentation. I wanted to ask you a little bit about the working on the reefs in Lake Erie, and I was curious if it seems like there's a lot of really great implications for reef restoration, and I was curious if that's something you guys are thinking about or like making recommendations about what kind of like where you are with that aspect of the work, because I know that's something like DEC is really interested in too. So anyways, just curious.
Yeah, so there are a couple sort of long-term implications for our findings. I think some of them have to do with how we're raising fish in the hatchery or where we're stocking them, the strains that we're stocking, and they've been very adaptable to a lot of that information so far. I think there are additional bits of information that can be useful to those activities from our work as we sort of develop, but definitely the specifics of where a reef is located, how does it interact with the hydrology, what does the physical makeup of that look like? If we can, that's sort of the idea of our habitat suitability index that we're working toward, is if we know the conditions that are, make for magic for lake trout reproduction, then we can identify places that would maybe be sort of the low-hanging fruit, either for restoration or artificial reef construction. So we are closely working with the state organizations to sort of think about those long-term implications of what we're finding.
Cool. Did you have a question? Oh, I'm going to read, there's two online. One is from Terry. Any evidence of round goby becoming an important prey species for lake trout and other native species?
Yeah, so that's not something we have specifically looked at, but I know that's been found in other systems in the Great Lakes, that lake trout are eating round goby. I think there's some indication that round goby are also helping the thymine problem in some of our salmonids. So it depends on who you ask whether round goby is a good or a bad thing. But so certainly they're an important part of the food web in Lake Erie, particularly as a prey item. And there was just a really amazing Lake Erie food web paper out that included round goby as one of those sort of important diet items. So there is definitely good and bad to be had with the invasion of round goby. They do, I think, serve as prey item for some of the larger piscivorous fish in the tributaries as well. But most of what our round goby project was focusing on was the sort of smaller bodied fish because we were specifically interested in competition.
So I'm going to read the next online one because it's kind of like on the opposite end of the spectrum. And with this question, I just want to note that during Corey's PhD, she worked a lot with some of the local watershed groups, especially Friends of the Rouge. And I recognize this name as one of the Friends of the Rouge volunteers. It's Phil
Hi, Phil.
Kukulski. Yeah, sorry.
It is so nice.
Yeah, butchering that. But he wants to know, do you find broken Asian clam or zebra mussel shells in goby stomachs?
Yeah, that was a great question. We did not find any in our New York tributaries. We did find a handful of those when we did similar things in Michigan. But the abundance of both corbicular or basket clams and quagga mussels, zebra mussels in the tributaries in New York is very, very low. We do have a master's student from another lab who just finished her master's working on the bivalves that are specifically being consumed. Most of them are spherid mussels. They're native mussels. They're not unionids. But they at least in the tributaries that we've seen, there's been very little consumption of dreissenid or corbicula invasive species. Mostly they're eating the native ones when they do eat bivalves, but it is a relatively low proportion of their diet as far as we're seeing.
Cool. Yeah, we have time for one or two more questions. Anybody in person want to ask one? Okay. So we have one more online. Oh, it's from Sally Petrella from Friends of the Rouge.
Hi, Sally. Nice to hear from you.
Given what you have learned about the round goby's effect on darters, what are your thoughts about reintroducing rainbow darters into the lower Rouge system or other systems that have only Johnny darters?
Ooh, that's interesting. Actually, I might have an extra bonus slide that goes over this. So we did look at the differences between specific darter species when we had adequate sample sizes to do that. In terms of their competition with round goby, it seems like they're maybe doing totally different things. So this one on the left here, for example, is log perch versus rainbow darter. The rainbow darter had a much higher niche overlap with round goby than did log perch. But that said, it was relatively low at a different site. So there could be some additional like habitat context that's regulating which darter species are sort of feeling the bulk of this interaction. But it's a little tough to say with relatively low sample sizes of rainbow darter specifically how they would respond to a reintroduction if the round goby problem hasn't been curbed. Just because they're doing different things than Johnny Darter or log perch. So probably a dissatisfactory answer, but I don't totally know that we know enough to say whether or not a reintroduction program would work.
Cool. Last call for questions. Okay. Well, yeah, thank you so much, Corey. Yeah, just one last round of applause for Corey, and then we can conclude the seminar.
Thanks so much.